Right, hello. Welcome back to BIOL1309. Today we're going to talk about the evolution of vascular plants. We're going to cover quite a lot of things, including more life cycle as well as the basics of vascular plants. And obviously, first of all, we're going to get the xylem and phloem out of the way, the vascular systems. Last video, I briefly touched on the vascular system, so I'm not necessarily going to go through detail like here. Vascular system, essential for progressive occupation of land. You can look at it yourself. Yada yada yada. These are just blah 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 things. Okay. Xylem transport water and mineral. Phloem transport water and food. This goes one way. This goes two ways. Blah blah blah. Anyways, let's go into the meat and bones of this video, where we talk about the actual important bits. First, the xylem. It's kind of ironic. Anyways, so xylem functions upward transport of water and minerals, just like how I said before, and the structural support, the lignin, if you remember. And then the tissues, there are two kinds of tissues for xylem actually. We have the tracheas, this one, which is present in all vascular plants actually, and the vessel element, which is only in angiosperms. But angiosperms have both of these, okay? So let's first talk about this new thing, new to you, I believe, and to me as well. Tracheothetes, which are primitive kind of xylem vessels, so to speak. They have, instead of no end walls, they have some end walls, but they're pitted, so they act kind of like the sieve cells for phloem. Again, they're still used for transporting water, but it's kind of less effective. The vessel element is wider with no end walls and is more effective in transporting water. And also, if you should know by now, both of these are made of dead cells and they are hollow as well to um, have less resistance when it comes to transporting water, as well as I hope you know how the method, how like the water is pulled up. The force is called transpiration pull, where the water is evaporated from the leaves and then there's a pulling force of the water because of capillary action and like pressure difference and whatever. All right, moving on to phloem. So phloem functions in terms of, well, in transporting water and food. It can both go upward and downward. Xylem only goes upward. All right, so we have in seedless vascular plants, they have sieve cells. These are sieve cells, one long strand. Sieve areas are kind of like areas that allow the cell and another adjacent cell to have mineral, no, not mineral, material exchange. So, for example, maybe there's another sieve cell here, and this cell would use the sieve area to maybe move some sugar, and then it will allow the sugar to exit from this cell and to the cell next to it. That's how the sieve area works. And for the sieve tube, which is only present in angiosperm, is more advanced. Instead of just having like one long strand individual cells, it has cells that are connected to each other. Ooh. And then they have sieve plates, which are just like end walls. And most effectively, they have something called the companion cell, which is attached to the sieve cell. The companion cell help like carry out cellular activities and like give food and give oxygen, remove waste from the sieve cell, such that the sieve cell could focus on um, transporting food and water, as well as removing like the important organelles out of the way, so that the tube is still hollow, and it could allow effective transport. This is the, yeah, that's how the sieve tube element works. Right. Did I miss anything? I don't think so. I think that's all the important bits that I have to go over for you. Oh yeah, so this is just wider. I think I just wrote down. Okay, so now this is the steel arrangement. So this is not the stele, it's the steel. The steel arrangement is the how the xylem and phloem is arranged in primary vascular systems. There are two types. There's the protosteel and the siphonosteel. The protostele means that the solid core of this vascular tissue. So it is in the middle. The xylem and phloem is the core of the stem or whatever. Yeah, but the steles means that the xylem and phloem are wrapped around the core. So they're not the core, but they surround the core. All right. So let's first talk about protosteles. Protosteles, three kind. One's a very simple xylem inside, phloem outside, circular kind. And one more thing. So xylem is always represented red in diagrams because through staining using I think like red stain or what stain, only through staining xylem always shows up red. So it's red. Phloem is blue to just give contrast. All right. So the second kind here, which is a cross shape for the xylem, but again still surrounded by the phloem, and the third kind, which is the xylem kind of interspersed in the phloem. I'm not too sure how to describe this sort of arrangement, but so yeah, just in the phloem, there's lines of xylem. Yeah. All right. So that's all the three kinds of protosteles. Now we move on to symphonosteles, which means the xylem phloem are surrounding the core. Right. So type one of siphonophils, which is the amphiphylic siphonosteles, which means that the xylem is surrounded by the phloem. You see, no matter it is a continuous ring of circle or this bundle, the xylem is always surrounded by the phloem. Okay, and the second type is the ectophilic siphonosteles, which means the phloem is outside while the xylem is inside. Always the xylem points towards the core, okay? No matter what. So this one may be bundled up very nicely on the, I would say, like perimeter, but this one is like interspersed, randomly put. But you can see the xylem always faces the core, no matter what, and the phloem always faces outside. So this is ectophloic cephalnosteles, and this is amblyophotic cephalnosteles. All right. And then, so the evolution of the steels. First, obviously, the proto-elite steel came first, while these two are evolved independently from each other. And these two are more advanced. Now, this part. The complexity of the plant body is truly complex. I would need some time to explain this. Alright. So this is kind of just why are like some parts that may be like on the liverworts, there might be like leafy structures, right? But they're not leaves. That's because leaves are leaves because of their vascular structure or vascular, they have like vascular tissues on them. That's how they're called leaves. Otherwise, those are not leaves. Those are just leafy structures. All right. Similarly, roots and stems they also have the same thing. All right. For a leaf, there are two types. We're going to the real bones here, like on the bone marrow type of horribleness. We were just on the meat, now we're on the bone marrow. So we have the microfilm. This thing, they're very small compared to the next one, and they have a single strand of vascular tissue. So this is the protostele, and then inserting, well, going through the microfilm, like one single strand of vascular tissue, while the megaphyll, the bigger leaf, large with expanded leaves, and they have complex arrangements of branching veins. So you can see this is kind of like this is the siphonophil, where the xylem, phloem, the vascular tissues are on the outside, not the core, so it surrounds the core. And this part, the vascular bundle or the vascular tissues, go through the leaf and then more branch out. So the branching veins. Leaf types. Okay, this is what I was talking about. So microphyll associated with protostele. The black stuff is the xylem phloem, and you can see this is kind of like a poor representation. But anyways, just remember microphyll, protostele usually, while megaphyll is cephalostele complex. Again, this part is just how they represent stephanosteles, that it is wrapped around the core on the perimeter, but this diagram is quite hard to understand. So just remember these two statements. That should be fine. Okay, a bit of an evolution on how the microfills are formed. Originally, the plant, not sure if you can call it a plant back then, but wait, no, you have the vascular tissue. So yeah, plant. The plant only had they had no arms or limbs, no leaf, and then a structure kind of grew. As it slowly grew, the vascular tissue continued to grow inside it as well, and then finally the vascular tissue is established in this leaf, and this is how microfills are formed. This is just the evolution. So originally it was this very long ago, and then as the microfill forms, it is evolved. Um, the vascular tissue evolves with it, basically. And this one is probably easier to explain. Like for megaphils, originally they were like highly branched branches of the plant, but some parts kind of devolved or shrunk and became smaller. But the xylem and phloem are still very like branched out and expansive. And then, but one single stem kind of grew into this central one. But and then so you had to end up with this. This is kind of like a imagine this. Imagine this leaf only having veins and no other structures. So only xylem phloem on the leaf, but without like the rest of the leaf structures. This is kind of like what it happens. And then as it grew, more of the leaf structures, the photosynthetic parts grew in. So that's how the leaf, the megaphyll, have an extensive vascular tissue of the veins and such. And then the leaf types. So primitive condition with no leaves, and then they grow independently from each other. All right, this is another very exhaustive part. Jesus. Okay, so remember how in non-vascular plants, the dominant individuals are actually gametophytes? For vascular plants, it's completely different. The dominant individuals are sporophytes, the ones that produce spores. So they started to use less water. Well, they started to they don't really need water for reproduction anymore, and sperms and spores are no longer really flagellated. But later, like some other examples, the first examples of seedless vascular plants, they still need water for sperm, I believe. Or at least the sperms are flagellated. Spores are not flagellated long ago, I think. So, and then this is the really silly part: progressive reduction of gametophytes. Remember how in like algae or whatever the liverworts and such, the gametophytes actually help nurture the sporophytes. Now it's completely different. The sporophytes are nurturing the gametophytes, and the gametophytes are actually becoming smaller and smaller. Crazy. And then finally, you have homospory and heterospory, which I will talk about later. Again, the cycle showing that the sporophyte is dominant. Remember, sporophyte is diploid. All right, homospory. So homospory, which means just same spore. So that means the sporophyte would produce spores. So only one kind of spore, and that spore would grow into one type of gametophyte, which is usually bisexual. So this gametophyte is able to produce both the egg and the sperm, and they could either maybe do self-fertilization or cross-fertilization to produce the zygote and grow into the sporophyte. For heterospory, the sporophyte actually forms two types of sperm. One is the microspore, and no, not sperm, sorry, the spores. One is the microspores, and the other is the megaspore. So the microspore would grow into the microgametophyte, which is the male gametophyte, you could say, and it produces sperm. while the megaspore would grow into the megaspore gametophyte, which produce the egg. This is just female. It's kind of like just male and female, really. It's a fancy name for it. And then fuse to form the sporophyte again. And this is a primitive condition for homospory, and the advanced condition as the heterospory. So there are advantages for heterospory in terms of evolution. The spores develop into unisexual gametophytes, so they only have one sex. All right. This encourages cross fertilization, which helps increase the genetic variation among the population and allows like natural selection for individuals to cope with adverse conditions. All that stuff, yada yada yada. And then finally, actually no, it's not finally. Shoot. In heterosporous plants, gametophytes are endosporic. Endosporic means the gametophytes retained inside the spores. And again, some primitive condition gametophyte that are independent of sporophyte, and then to the gametophyte being inside the spore. 1-to-1 correlation. I don't think there is any difference, like not that any difference, any exception to these two statements. All plants with heterosporous life cycle have tiny endosporic gametophytes. So all these plants that are heterosporous, that forms megasporophyte, no, megagametophyte or microgametophyte, the spores or the gametophytes are always within spores. While plants with homosporous life cycle, they have still small but comparatively larger free-living gametophytes. All right, and then finally some terminology. Microspore, megaspore means just male spore, female spores. Microspore born in microsporangia, blah 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 blah. And then microspore germinate to form microstrip. Alright, and here. So micro and mega have different meanings now. For in terms of leaf, microphyll just means small leaf. Megaphyll means big leaf. But in microspore and megaspore, it means male and female. Alright. So that is finally the end. I hope I have explained most things clearly, and if I haven't, you might need to use a bit of your brain power. I have faith in you. So yeah, that's really it. It is quite a sudden jump in difficulty, I would say. Like we were talking about, oh look at this tiny little plant, and then suddenly explosion, bang, microgametophyte and megagametophyte, boy. Yeah. So these will be the basics. You will see a lot more of this later because we're going to the realm of vascular plants. So clear the concept up so that you're not making yourself dizzy as you go through the rest of the lessons. All right. All right. That's the end from here. That's the end of me here. I'll see you in the next video. Bye bye.